Okay. Hi guys. So today we're going to continue from where we ended last time. So today we're going to be looking at the topic which is known as digestion and absorption. Okay. So now, when you look at digestion and absorption, you're going to realize that we have got at least three basic macromolecules that we're going to be discussing. Okay. So we're going to start with what we call carbohydrates. Then we'll go to lipids or proteins. Okay. So these are the three basic macromolecules that we are going to be breaking down. Okay. So now, when you were back in high school, you learned about this concept called digestion, or this topic known as digestion. Okay. So now, digestion does not only apply to what we call chemical digestion. So it has got what we call physical digestion and also chemical. All right. So in high school, you used to learn about the physical aspect of digestion as happening only in the mouth. But you're going to realize that now, when you talk about the concept of digestion, you should have a broader perspective of view, because the physical aspect of digestion does not only happen in the mouth, or it's not only contained in the mouth, it goes all the way. Okay. Yeah. So when you talk about mastication, that's a physical aspect of digestion, right? The churning in the stomach, that's physical aspect of digestion. When you talk about peristalsis, that's also a physical aspect of digestion. So you're going to learn that as we go on, these same concepts are going to also be involving the reducing of the particles into very small minute particles, so that enzymes and other gastric juices or other um, alimentary canal juices can be able to degrade. All right. So before you go to this topic, I believe you did go through what we call secretions. Okay. So this comes after secretions. So you're supposed to know the different types of secretions which are produced by the alimentary canal. All right. So usually you're going to realize that most of the secretions are produced by what we call accessory organs. So these are organs that are not directly involved into digestion, but they act as helper organs. They help to produce these chemicals, which can be able to either degrade or inhibit or stimulate these chemical mechanisms. All right. So let's start with what we call the definition of digestion, and then we go to the definition of absorption. So what comes into your mind when you hear the word digestion? All right. So now, the first point is saying digestion is a process essential for conversion of food into small and simple form. All right. So the reason why the definition is as basic and simple as it is right now is because we are looking at two aspects of digestion: the chemical aspect and the physical. And the physical aspect. Okay. So now, when you look at the physical aspect of digestion, what do we mean? What does it involve? Okay. So now, the physical aspect of digestion involves like the things that I mentioned. So number one, the first point is saying it involves what we call mechanical digestion by mastication and swallowing. All right. So they're not wrong by mentioning those things. So when you refer to mastication, we are referring to the chewing action of teeth. All right. So when the food enters the mouth, all right. So when an individual goes maybe out to buy pizza or maybe Nando's or anything that they're supposed to consume, okay? So in the example that we can give is that we need to choose something that contains carbohydrates, lipids, and proteins, okay? So no wonder I'm giving an example of pizza. So you'd find that most of the time pizza contains all these three essential nutrients, all right? So now, when you're consuming, you get probably a piece of pizza, you put into your mouth, right? So the first thing that's going to happen there, you're going to find that the teeth will start to break down, okay, or degrade these complex substances. Okay, so it will start now breaking them down or grounding them. So that grounding action or the breaking down is known as mastication. So it's not easy for you to chew food without the help of the tongue. So the tongue also serves this purpose of trying to reduce the surface area for these particles by also producing different types of enzymes that are found in the various salivary glands. Remember, when you learn endocrinology or even maybe GIT, we need to know that there are different types of glands that we have in the body. There's what we call exocrine, and this is what we call endocrine. Okay, so the exocrine, these have got what we call ducts, and these belong to the class of digestive. They belong to the digestive aspect. Okay, then when you talk about endocrine, this has to do with the endocrine system, which has to produce what we call hormones. So these are known as ductless glands that produce hormones. And these hormones are responsible for inhibiting or stimulating certain chemical reactions, or in order to control what we call homeostasis. But we are not talking about that now. But as you can remember, when you are looking at secretions, we did talk about some examples of chemicals that belong to the endocrine system. Okay, yeah. But let's focus on what we are doing right now. Okay, so now so mechanical digestion by mastication and swallowing. And when you look at swallowing, if you remember what you learned under motility, swallowing has got 3 phases, huh? And you're supposed to remember all those phases. So all those are aspects of what we call physical digestion. Okay, yeah. Then the chemical digestion aspect. So this chemical digestion comes in. So you can even define it. This is the breaking down of large complex food particles into smaller ones with the help of digestive enzymes. So these are enzymes that are able to catalyze or speed up the process of breaking down these complex food particles, so that you can either get what we call monomers or, yeah. So the major point is to get those monomers so that you can be able to group them into what you can be able to use at a later stage, or so that they can be absorbed or assimilated. Okay, so now let's go to what we call absorption. What is absorption? Okay, so now after the process of degrading or reducing the surface area or breaking down, so you get what we call monomers. So you're going to find that under carbohydrate digestion, you're going to get what we call fructose. Sorry, you're going to get fructose, galactose, and glucose. So these 3 monosaccharides are supposed to be taken up by the body. All right, so they can only be taken up by the process called absorption. All right, so there are specialized cells along the alimentary canal or the GIT tract which are able to allow these monomers to be taken into the bloodstream. Okay, because they are needed. As you know, glucose is one of the key elements that's needed under what we call respiration to produce what we call ATP. To be more precise, under aerobic type of respiration, although we have got what we call anaerobic. That's the major factor. Okay, so now we need to know how can we reduce these particles. I mean, how can we reduce these foodstuffs, or how can we reduce these macromolecules into micromolecules or monomers? Okay, that's the purpose of digestion. Once you achieve that purpose of digestion, then now these monomers can now be taken up, assimilated, or absorbed by the body. So now this below is saying absorption is the process of transporting small molecules from the lumen of the gut into the bloodstream and lymphatic vessel. So when we talk about the bloodstream, you know that we have got macro molecules, monomers from carbohydrates and proteins are going to be taken up by what we call the bloodstream. But those which are coming from the lipid family, the monomers are going to be taken up by what we call the lymphatic system. All right. I think the reason why they're going to be taken up by the lymphatic system, I think we did cover this under what we call cell physiology. So now, since you already have an understanding, or we have introduced what we are going to look at, let's go to the next slide and be able to appreciate the information. Okay. All right. So now this diagram is just something that you have been seeing for a long time. Okay, in your previous lectures, also when you're in high school or when you're doing maybe A levels or something like that. So now this is what we call digestion and absorption, right? So now look at this diagram. It's showing the different areas which participate in what we call digestion and absorption. So if you can see it saying input, that's the mouth, that's where you take in. You don't get food via the nostrils, unless maybe you are sick. Yeah, that can be discussed under pathology. Yeah, but basically, since physiology is the study of the normal function of the body, so the ideal area where you're supposed to take up something is the mouth, which is known as the buccal cavity. So food and drinks can be able to go through there. Even air can also go through there. So you can see here they are saying that we have got what we call salivary glands. So these are about 3 salivary glands that are responsible for releasing enzymes that can be able to start digestion there. Okay. Then after that, there's what we call the esophagus. There's also the pharynx up there. So pharynx, then esophagus. Then you can see that we have got this accessory organ, which is known as the liver. So they are saying that it produces what we call bile. Okay. So bile, you know that it's a very essential molecule in the breaking down of lipids. All right. Yeah. Then most people mistake, they think bile is produced by the gallbladder. The gallbladder is just for storage, but the major organ that produces bile is known as the liver. Then there's what we call the stomach. Okay, so from the esophagus, you are going to have an adjacent organ which is known as the stomach. So the stomach has got about 4 parts, okay, which is known as the cardia, fundus, body, and pylorus, as you discussed back then under anatomy. So now this part is responsible, okay, is responsible for breaking down proteins and also some small chained lipids. Okay, so as you know, there are some under secretions, as you learned. There are different types of juices that are, there's what we call gastric juice, which contains different types of chemicals, all right, which includes pepsinogen, which gets activated to what we call pepsin by what we call hydrochloric acid. So then after the business in the stomach is done, then near the pylorus, there's what we call the pyloric sphincter. So this pyloric sphincter is responsible for opening up and emptying the gastric content or the stomach content into the duodenum. So after the duodenum, you're going to find that we have what we call the jejunum. Then jejunum, then there's what we call the ileum. Then ileum, we're going to join what we call the ascending colon. Then there's what we call the transverse colony, then a descending, etc. So now you can see here they are saying we have got what we call the pancreatic secretion. So the pancreas are responsible. The pancreas contains what we call exocrine cells, which are known as the acini cells. So these acini cells are able to produce different types of chemicals that can continue the digestion started in the mouth and in the stomach, right? For different types of substances. Yeah. So once that happens, those chemicals or those enzymes and other different types of chemicals are going to be emptied into what we call the duodenum. And once that happens, then digestion will continue, right? But we'll go in detail as we move on. Okay. Yeah. So now, or maybe I can use this diagram to explain. Let me just check how much, what is there? Okay, so I think digestion, we can even just use this same one. All right. So now, what we have here, if you can see, there's pancreatic secretions there, there's intestinal secretions from small intestines, from large intestines, then there's excretion in feces, removal from the lumen. Yeah, so this is a bit of more like a summary of what you covered under secretion. So basically, so if you remember very well, when we were talking about carbohydrate digestion, okay, so it's known that carbohydrate digestion starts in what we call yeah. So look at this diagram. So input. So I gave an example of what we call pizza. So if you ingest pizza here, right? Yeah. So in the mouth. So food and drinks, depending on what you're taking. So as you know, pizza contains, usually whichever type you get, it is going to contain what we call carbohydrates, which is starch. It's also going to contain lipids, right? Which might be in form of cooking oil or anything that you took. Yeah. Then proteins, in case maybe there is cheese or there is some chicken, etc. So that would be a protein source. So now, when you put food in the mouth, there's what we call digestion of both starch is going to start here, all right? So the moment it begins in the mouth, the salivary glands are going to produce what we call saliva, okay? And these are known as accessory organs. So once they produce that, they contain they contain salivary amylase or ptyalin. They also contain there's what we call lingolipase. They also contain bicarbonate and other things, okay? So now, basically, our major focus is to look at digestion, right? So now, the moment the enzymes are produced, so for example, if the individual or the person, okay, or the subject took starch, okay, in the pizza, the starch, so there will be mastication happening there. The food will be ground continuously. So as the food is being ground there, okay, it's being constantly mixed with saliva with the help of the tongue, right? So as the mechanism is happening continuously, the mixture is happening. So also digestion will begin, right? So now you're going to find that there are very few people who can chew food for a minute. Usually it's just a few seconds, then you swallow. But the enzymes would have mixed sufficiently with the saliva, okay? Meaning digestion has already begun. So the moment that begins, the food is going to be now swallowed. So in the mouth, you're going to realize that we are going to have starch, which is going to be broken down into what we call maltose, which is a disaccharide, oligosaccharide, and dextrins, all right? So dextrins contain about 3 glucose molecules, right? Yeah, oligo, 3 to somewhere 10, depending on the book that you're using. So once you get that from starch, since starch is a polysaccharide, okay? It's a complex polysaccharide. So once you get that, then for lipase, it's also going to act on what we call small chained lipids and fat soluble vitamins, all right, to get what we call fatty acids and also glycerol. So, but the complex lipids, nothing is going to be done. So then after that, the bolus, which was formed due to the rolling action or the mixing action of the tongue together with the help of the teeth, then it's going to be pushed back by the tongue due to the 3 stages of swallowing, okay? Where the hard palate is going, the tongue is going to be elevated, then pushes the bolus against that palate, then the food will go down at the back of the pharynx, then after that, it's going to be ingested, okay? So, you know the 3 stages of swallowing, huh? That's your own baby toenails because we already covered that part. So now, then after that, we have got what we call the epiglottis, huh? So this prevents or protects the airways, all right? So that food does not go into the airways or the trachea. So it prevents choking. Remember, at this particular stage, the moment the food reaches the pharynx, starts going down, it's under what we call the autonomic nervous system, which is part of the enteric nervous system, right? So the enteric nervous system, as you know, is the concept that deals with GIT nervous system, all right? It's also known as the brain gut, okay? Or the gut brain. Sorry, I've mixed them. The gut brain, meaning the brain of the stomach, the gut. So that's known as the enteric nervous system, which has got 2 parts. There's what we call the intrinsic and extrinsic. All right. Yeah. So now I've got also detailed notes concerning this, which I've already forwarded to your group. But so after that, there's what we call also the upper esophageal sphincter, which prevents air from entering the esophagus, right? Yeah. So after that, there was going to be what we call peristaltic movement. Remember what you discussed in the anatomy, that the rings of the esophagus that are made up of what we call circular and longitudinal smooth muscles. So these muscles are known as antagonistic, which causes the peristaltic wave, which pushes the food down the esophagus. I always give this example to my students that the best example that you can know this wave-like motion is when you take sweet potatoes that have got a lot of starch and you don't have water. You're going to feel how this is going to be moving until it reaches the stomach. So before it reaches the stomach, there's another sphincter there known as lower esophageal sphincter, which prevents regurgitation or prevents you puking up the things that are in the stomach. It is a tight muscle that prevents regurgitation, meaning the flowing back of food into the esophagus, which might cause what we call heartburn. All right. So now, after that, the food will reach the stomach, or the bolus will reach the stomach. So once the bolus reaches the stomach, in the stomach, all right, we have got what we call gastric juice produced by different types of cells. Okay. So this gastric juice contains three important things. Number one, it contains hydrochloric acid. Number two, pepsinogen. Number three, mucus. So these three things are important. Number one, to be able to, I'm not saying those are the only things that are found there. These are just the important things that you're supposed to take note, but I'll also mention the other things that are found there. So you're going to find that in the stomach lining, the mucus protects the stomach lining because there is an acid that is produced by what we call the parietal cell, which is known as hydrochloric acid, and it has got a pH of 2 to 3.5, somewhere there. It's a very strong acid. It just follows sulfuric acid. Okay. So now, once the food reaches the stomach, or the bolus reaches the stomach, there's going to be what we call churning, or there's going to be what we call gastric peristalsis, meaning the stomach will be moving in a wave-like motion, trying to reduce the surface area of the bolus and also to mix it with what we call the gastric juice. Remember where we're coming from. The stomach, the mouth has got a pH of, which is alkaline in nature, right? While the pH of the stomach is acidic. So the moment the salivary amylase reaches the stomach, it has to be denatured. So the moment it gets denatured, but this does not happen instantly. It takes about an hour or so. So this is where most of the carbohydrate digestion takes place. It takes about an hour or so. Because an hour compared to the few seconds, the open gullet that you do when you are consuming food. So it spends more time there in terms of digestion of carbohydrates. So the moment the churning or gastric peristalsis continues, it causes the mixing, hence causing the salivary amylase to be denatured, hence stopping the digestion of carbohydrate, hence commencing what we call protein digestion. Remember, like I said last time, the stomach is specialized to degrade one specific type of protein, which is known as collagen, that elastic type of protein. Okay, so if the stomach has got challenges in terms of digestion or in terms of secretion of pepsin, it affects what we call collagen digestion. So once pepsin is produced, pepsinogen is produced and converted to pepsin, it's going to go and act on the protein to convert them into what we call peptones or polypeptides, right? Like I said last time, proteins are the most difficult to break down as compared to the other type of macromolecules. Then there's also another culprit that is found in the stomach known as gastric lipase. So gastric lipase is also going to have the same function as what I had explained in the mouth, okay? So the mouth and the stomach account for like 10% of lipid digestion, okay? So you can say after the mouth, the stomach has got about three things happening: protein digestion, lipid digestion, and carbohydrate digestion. So then after that, as the churning continues, the food is being degraded and reduced in terms of size. Then there's going to be a mechanism or a chain of reaction that's going to cause what we call gastric emptying. I know under the other things that I've not mentioned, gastrin, I've not mentioned somatostatin. Yeah, but that's, you're a student, you can go and research and study more about that. All right. So now, After that, we are going to have what we call gastric emptying. So gastric emptying is the opening of the pyloric sphincter to the duodenum, hence allowing the chyme or the stomach contents to be emptied into the duodenum. Remember, the stomach content is acidic in nature, all right? So there's going to be a mechanism which is going to be caused by CCK, okay, which is going to cause the release of bicarbonate from bowel. The release of bicarbonate, which is found in bowel, which was stored in the gallbladder, and also which is also going to cause the secretion of different types of enzymes from this part, which is known as the pancreas. Right? So once those enzymes enter the duodenum, for example, for carbohydrate digestion, we have got what we call pancreatic amylase. So that pancreatic amylase is going to continue what started in the mouth and ended in the stomach. Remember maltose, dextrins, and oligosaccharide, right? Then we're going to have, for lipids, I can go maybe to lipids so that I can be able to complete as soon as possible. So now when you go to lipids, you're going to find that under lipids, there's going to be what we call different types of enzymes going to produce. So remember, so there's also going to be what we call phospholipids, A2, phospholipase A2 is going to be produced. Then there's also going to be cholipase, which is a core enzyme. Core means helper enzyme. Then there's also going to be pancreatic lipase, all right? And there's also going to be about 4. There's also going to be what we call cholesterol esterase, okay? So these are the 4 different types of enzymes that work in order to degrade the lipids. So from the names, you can say that cholesterol esterase has to do with cholesterol ester, which produces what we call cholesterol. Then phospholipase A2 is going to give us phosphate and fatty acids, all right? Then for triglycerides, all right, they're going to be acted on by what we call pancreatic lipase. So this pancreatic lipase cannot anchor, all right, to the triglycerides without any help. They need what we call cholipase. So before all this happen, they need what we call bile. So bile helps into what we call emulsification. So what is emulsification? So emulsification is defined as the breaking down of large complex fatty droplets into smaller ones. Okay, how does that happen? So when you talk about bile, bile contains lecithin, contains also bile salts. So these bile salts and lecithin, they are hydrophilic, meaning they're water loving, while fats are hydrophobic. So they convert fats from being hydrophobic into being hydrophilic, into small tiny droplets. Hence, what I mean by that is that they create a coat on the outside, which is hydrophilic, but hydrophobic inside, so that they can create that interaction between the water molecules and the fatty droplets, because it will be difficult for you to degrade or digest something that does not interact well with water once it's there. So it needs to first to be converted. Just like, I'll give an example. If you want to marry a Jehovah's Witness, you need to first become a Jehovah's Witness. Then you marry that person. Okay. So even fats go that conversion, okay? They need to be hydrophilic. And what helps that? It's known as bile salts and lecithin. So these help in terms of trying to cause what we call the molecules being hydrophilic. So once they're hydrophilic, so they can interact, the water is able to interact with the fatty droplets, and also the enzymes, as you know, enzymes are hydrophilic in nature, meaning they're water loving. So the enzymes which are water loving and the fats who are pretending to be water loving, then they can interact. But again, anchoring of the droplets and the enzyme is difficult. So we need a helper enzyme, which is known as cholipase. So cholipase is going to anchor on the fatty droplets and also anchor on the pancreatic lipase. Then start up the digestion, which we are going to produce what we call glycerol and fatty acids. So once we have all the contents of the lipids, formed. Then there's going to be what we call a micelle. A micelle is going to contain all those things that I've mentioned, even fatty soluble vitamins. Then after that, then they can be safely transported. Remember, these materials are not water loving. So they create what we call a micelle. So this micelle can now be transported to areas in the jejunum and the ileum where they can be peacefully absorbed or assimilated. All right, into what we call the lacteal, because the lacteal is an extension of the lymphatic system. All right, so now for carbohydrates, you're going to realize that as they continue moving, since when you are consuming, remember it's saying drink, so drinks contain a bit of what we call sucrose, and some artificial sweeteners, etc. These cheap drinks. So now you're going to find that there will be sucrose there, there will be maltose there, there will also be lactose in case you took some milk. All right. So as you know, for sucrose has to be broken down into fructose and glucose by an enzyme which is going to be produced by the brush border enzyme, which is known as sucrase. All right. Then this is happening in the jejunum and ileum. Then for lactose, it's going to be called lactase. So people have got what we call lactose intolerance is because they don't have, or they produce less of what we call lactase. Hence, when lactase acts on lactose, it's going to produce what we call galactose and glucose. When maltose, which was one of the byproducts from breaking down of starch from the pizza, it contains two glucose molecules. So when you act on by the enzyme known as maltase, it's going to give us what we call two glucose molecules. Then for the dextrins, they're going to be acted on by what we call the alpha limit dextrinase enzyme, which is going to give us glucose molecules. Remember, the difference between oligosaccharide dextrins and maltose is due to the fact of 1 to 4 and 1 to 6 glycosidic bond. If you're able to remember under biochemistry, okay, how these bonds are formed. Amylose, amylopectin, I'm not here to teach biochemistry, but if you can go back to biochemistry, you're going to understand how a starch molecule is formed. All right, so like that. So when you break down the oligosaccharides, it's going to give us what we call glucose molecules. So once you get those are going to be transported to the brush borders where there are enzymes which can be able to assimilate or absorb, which is the next part which you are going to do. Then for proteins, they have got almost a similar pathway, but there's they've got a similar, but it's a bit more complex. You're going to find that there are about 4 types of enzymes that are going to be produced by the pancreas. There's going to be trypsinogen, chymotrypsinogen, there's going to be pro-elastase and pro-carboxypeptidase. So as the name differ, that's what they target. All right, that's what they target. So you can go and look at what they target. After that, they are going to produce what we call disaccharides. They're going to produce what we call dipeptides and tripeptides. So these tripeptide and dipeptides can go again, they can move into the jejunum and ileum where digestion will still continue. All right. So once those are produced, you are going to find that the brush border enzymes can produce enzymes that can be able to degrade dipeptides and tripeptides. So these enzymes are known as tripeptidase and dipeptidase. They can be able to degrade these further to form what we call amino acids. But you're going to realize that protein digestion continues up to inside the cells, where we're going to have again the same type of enzymes which can be able to degrade further. Then after that, the food will be able to move down into the rectum. Then after that, pieces can be passed on. Yeah. So after you know the simple simple mechanism that has to do with digestion and absorption, we can now go into what we call, so this is whatever I was just trying to discuss. So my major focus is this. So this diagram is just showing intestinal villi, surface areas. So it's just showing us the areas. These are the villi, the crypts. Endocrine cells, complete cells. Okay. So now this is the mechanism I was talking about, if you can be able to see. Yeah, so in your own free time, you can go through that. Yeah. So now that's carbohydrate digestion. Then here, this is how carbohydrates are going to be absorbed. So now, basically, if you're able to remember under cell physiology, how carbohydrates can be able to be absorbed, then it will be easier for you to be able to understand. So actually, we have got what we call a pump that is being used. Then there's also what we call a co-transporter of glucose, which is also being used, if you can be able to see there, right? Which has got sodium and glucose or galactose. So glucose and galactose use the same carrier protein. So this carrier protein has got a name, and this name is known as sodium glucose transport 1 or 2. Okay, so they've got the same mechanism where they allow, or where sodium and glucose are transported together. Remember, you said uniport, symport, whatever. Yeah, so this is known as a carrier protein. And at this particular level, you're supposed to know what a carrier protein is, what a channel protein is, and what a pump is. It's easier for you to be able to understand. So the moment glucose and galactose are taken up, so you're going to find that glucose and galactose share the same carrier protein. Yeah, then you're going to find that we have got what we call GLUT5. So GLUT5 is found on the apical membrane to transport fructose. It uses a different type of carrier protein. So this is known as facilitated diffusion. This is known as secondary active transport of glucose. Because on the basolateral membrane, this is known as the apical, and this is known as the basolateral membrane. So on the apical membrane, there's what we call secondary active transport happening. Then on the basolateral membrane, if you can see, there's what we call primary active transport happening. That's what is happening here. So here you have got what we call capillary. So whatever gets in there or gets transported inside, like the glucose and the galactose, it will go then to live by what you call the GLUT2. Then after that, it will go into the capillary. Then after it goes there, then can be transported to areas where they are needed. Yeah, so this is what they're just trying to describe. But this mechanism, you can understand it under what we call cell physiology. All right. So now, another thing that you can be able to appreciate as an individual is that the same mechanism happening here is what happens for amino acids. Just replace the glucose and galactose with amino acids, but they don't use a carrier protein known as sodium glucose transport 1 or 2. They have got different, because there are about 20 different types of amino acids. So they have got different types of carrier proteins, but the mechanism is similar. The only difference is on the glucose and galactose, where you need to replace with amino acids. Yeah. So digestion of proteins, this is the summary. Yeah. So you can go through that in your own free time. Then this is what I was talking about, to say once the proteins are degraded by the peptides are by di and tripeptides are going to be acted on by what we call peptidase and tripeptidase, etc., etc., all those things. So once they're broken down, they can also be absorbed through the same mechanism I was mentioning. So the other thing that you can also take note is that there's also what we call endocytosis, which has to do with small peptides can be absorbed like that into the cell. Then they can be acted on by these enzymes to produce these small amino acids, which can later be absorbed by the blood. But for lipids, like we said, when I was looking at that, it was saying that they need to go through what we call the lymphatic system. Okay. And I think this is the end of the lecture.